This is part 3 of lecture 11 of respiratory physiology. This information can be found in chapter 22 on pages 819 to 857. Transport of respiratory gases by blood. Let's start with oxygen. Oxygen is carried in the blood in two ways. 1.5% of it is dissolved in the plasma, and 98.5% of it is loosely bound to iron of the hemoglobin and red blood cells. Each hemoglobin molecule is composed of 4 polypeptide chains. Each polypeptide chain has an iron-containing heme group, and each hemoglobin can transport 4 oxygen molecules. Oxyhemoglobin, or HbO2, is basically the hemoglobin-oxygen combination. Reduced hemoglobin, or deoxyhemoglobin, HHB, is hemoglobin that has released oxygen. The loading and unloading of oxygen is facilitated by a change in shape of the hemoglobin. As oxygen binds to hemoglobin, hemoglobin changes shape, increasing its affinity for oxygen. As oxygen is released, the hemoglobin changes shape and causes a decrease in affinity for oxygen. Fully saturated, or 100%, is when all 4 heme groups carry oxygen. Partially saturated is when 1, 2, or 3 heme groups carry oxygen. The rate of loading and unloading of oxygen is regulated to ensure adequate oxygen delivery to the cells. The factors that influence hemoglobin saturation are the partial pressure of oxygen, temperature, blood pH, the partial pressure of carbon dioxide, and the concentration of BPG, which is known as 2,3-biphosphoglyceric acid. Students need to be familiar with this table. This is the oxygen hemoglobin dissociation curve. In terms of the influence of the pressure of oxygen on hemoglobin saturation, we are looking on the left-hand side of the y-axis, the percent of saturation of hemoglobin, and on the x-axis, the partial pressure of oxygen, with the right-hand side representing 100 millimeters of mercury and the left-hand side representing 0 millimeters of mercury in terms of the partial pressure of oxygen, and on the y-axis, with the top representing 100% saturation and the bottom representing 0 saturation. As you can see, the association is not linear, but is curvilinear. The partial pressure of oxygen heavily influences the binding and release of oxygen with hemoglobin. The percent of hemoglobin saturation can be plotted against partial pressure oxygen concentrations. The resulting graph, as we said, is not linear, but an S-shaped curve, and this is called the oxyhemoglobin dissociation curve. In arterial blood, the partial pressure of oxygen is 100 millimeters of mercury, and contains 20 milliliters of oxygen per 100 milliliters of blood, or 20% volume. Hemoglobin is 98% saturated. Further increases in the partial pressure of oxygen, as in deep breathing exercises, produces minimal changes or increases in oxygen binding. In venous blood, the partial pressure of oxygen is 40 millimeters of mercury and contains 15% oxygen. Hemoglobin is 75% saturated. The venous reserve is the oxygen remaining in the venous blood that can still be used and can be represented by this amount. As you can see, once we get past 75% saturation, or below 40 millimeters of mercury, there is a steep decrease in percent saturation. This is why, in resting conditions in hospitals, there is not much concern if oxygen saturation remains in the high to mid 80% zone or above. Anything below 80% saturation can be of concern. In terms of the factors influencing hemoglobin saturation, an increase in temperature, hydrogen ion concentration, or a drop in pH, an increase in partial pressure of carbon dioxide, and an increase in BPG can modify the structure of hemoglobin. This results in a decrease for hemoglobin's affinity for oxygen and occurs in systemic capillaries. However, this enhances the unloading of oxygen, causing a shift in the oxygen hemoglobin dissociation curve to the right. A decrease in the factors will shift the curve to the left. and decreases oxygen unloading from the blood. BPG is produced by red blood cells during glycolysis. BPG levels will rise when oxygen levels are low, and as cells metabolize glucose, they can use oxygen, which causes an increase in partial pressure of carbon dioxide and hydrogen ions in the capillary blood. Declining blood pH, that is acidosis, and increasing partial pressure of carbon dioxide cause the hemoglobin-oxygen bond to weaken. This is referred to as the Bohr effect. Oxygen unloading occurs where it is needed the most. Heat production in active tissues would directly and indirectly decreases the hemoglobin affinity for oxygen and allows increased oxygen unloading to active tissues. Carbon dioxide transport. Carbon dioxide transport is a little bit more complicated than oxygen transport. Of carbon dioxide transported in the blood, approximately 7 to 10% is dissolved in plasma as carbon dioxide. 20% of carbon dioxide is bound to the globin part of hemoglobin and produces carbaminohemoglobin, and 70% is transported as bicarbonate ions, or HCO3 negative, in the plasma. It occurs primarily in red blood cells, where the enzyme carbonic anhydrase reversibly and rapidly catalyzes this reaction. In the systemic capillaries, after bicarbonate is created, it quickly diffuses from the red blood cells into the plasma. And this outrush of bicarbonate from the red blood cells is balanced as chloride moves into the red blood cells from the plasma, and is referred to as a chloride shift. Here we can see a diagrammatic representation of the transport of carbon dioxide. The left-hand picture demonstrates carbon dioxide being picked up at the tissues. The right-hand side represents carbon dioxide being released from the red blood cells and from the plasma into the alveoli. In pulmonary capillaries, the process basically occurs in reverse. Bicarbonate removes into the red blood cells, while chloride moves out of the red blood cells back into the plasma. Bicarbonate binds with a hydrogen ion to form H2CO3. H2CO3 is split by bicarbonate anhydrase into carbon dioxide and water, and the carbon dioxide diffuses into the alveoli. Haldane effect. Haldane effect is referring to the amount of carbon dioxide transported that is affected by the partial pressure of oxygen. The lower the partial pressure of oxygen and hemoglobin oxygen saturation, the more carbon dioxide can be carried in the blood. Reduced hemoglobin buffers hydrogen ions and forms carbaminohemoglobin more easily. The process encourages carbon dioxide exchange at tissues and at the lungs. At tissues, as more carbon dioxide enters the blood, more oxygen dissociates from the hemoglobin, the Bohr effect. As hemoglobin with oxygen releases the oxygen, it more readily forms bonds with carbon dioxide to form carbaminohemoglobin. The influence of carbon dioxide on blood pH. Carbonic acid and bicarbonate buffer system helps the blood resist changes in pH. If hydrogen concentration in blood rises, the excess hydrogen ions is removed by combining with bicarbonate to form H2CO3, which dissociates into carbon dioxide and water. If hydrogen ion concentration begins to drop, H2CO3 dissociates, releasing hydrogen ion concentration. Bicarbonate is considered the alkaline reserve of the carbonic acid bicarbonate buffer system. Changes in breathing rate and depth affect blood pH. Slow, shallow breathing causes an increase in carbon dioxide in the blood, resulting in a decrease in pH. Rapid, deep breathing causes a decrease in carbon dioxide in the blood, resulting in a rise in pH. Changes in ventilation can help adjust pH when disturbances are caused by metabolic factors. Therefore, breathing plays a major role in acid-base balance of the body. And this is the end of this part of Lecture 11, our respiratory physiology.